My name is Nuran Zaghloul, and I am the NIDDK co-chair for the AMP and Common Metabolic Diseases. I'm joined up here by our panelists and by Melissa Miller from Pfizer, who is our pharma co-chair for this partnership. So we are excited to spend the next hour or so telling you about some updates from the AMP and CMD. But first, a little bit of context, a little bit of history. So the AMP and CMD was built on the success of the previous AMP in type 2 diabetes, which was, as we heard this morning, started out as one of the original disease areas for the AMP partnerships 10 years ago now. And the goals of the AMP in T2D were to bring together really the massive amounts of genetic data that had been generated by the type 2 diabetes community and make them accessible and usable to the broader research community in ways to really propel the field forward. And so to do so, the partnership created the Type 2 Diabetes Knowledge Portal and later on the accompanying Diabetes Epigenome Atlas, which really brought together very, very large amounts of genetic and other omic data from a very large number of studies and really culminated in data from over 1.5 million individuals. And then in addition to that, these portals generated tools for powerful analysis that were really tailored to the needs of the data in the portals. And so overall really became much more than a data repository, but really a one-stop shop for hypothesis generation and multifaceted analysis to move the field forward. And so with this really wonderful success, the stage was set to meet the next challenges, the first of which was to expand the type 2 diabetes success story across the metabolic disease space. And so that meant taking much of what had been done in the previous round and extending it to beyond diabetes and prediabetes, but to other related conditions like obesity, liver disease, kidney disease, and cardiovascular disease. And so the vision for the AMP and CMD was born with the creation of the CMD Knowledge Portal and the CMD Genome Atlas, and really has been supporting integrating existing as well as generating new data sets from across the omic data types in ways that are really quite powerful. So I'm gonna turn it over to Melissa to tell us a little bit more about the portals and where they stand.
Thanks, Nuran, and thanks everybody for being here. So as Nuran just mentioned, we have a couple of different portals in AMP CMD, and you'll hear more actually about both of these in subsequent talks. Noel Burt is going to talk about our Genetics Knowledge Portal tomorrow, and Kyle Galton, who is just sitting here next to us, is going to talk about our Epigenome Portal. But just as a high-level overview for our Genetics Portal, this portal has existed now for, I think, 7 or 8 years, and Noel could tell us the exact specifics on that tomorrow. But right now we have over 500 different data sets, and you can see that we have data sets from multiple different tissue types and also multiple different phenotypes. So in addition to having sort of things that we would consider common metabolic disease phenotypes, we also have summary statistics for genetics in there on other traits, things like retinal traits or sleep traits. So you can sort of look at the genetic relatedness of things like that with your traits of interest. In addition to that, we also currently have data. Noelle and her team have also managed to get investigators from 50 different countries to deposit their summary statistics, which is pretty amazing. So for the rest of the session today, what we're going to tell you about is some of the new data that we are generating for the CMD program. And in addition to really focusing on all of these different diseases, one of the things that we realized, we have a lot of genetic data. So what we've really focused on in the last couple of years is generating more single cell expression data and open chromatin data to be able to help us sort of annotate the loci that we're finding from genetics. So with that in mind, our first talk will be from Kyle, and he will be telling us both about the Epigenome Portal as well as how we use that portal to be able to annotate risk loci. And then we've got 2 additional talks from 2 of our large-scale projects that are generating data. The first one will be from David Brenner, and he will be talking about a multi-omics approach to metabolic liver dysfunction. I don't trust myself to say steatohepatitis on the stage. And then the next talk is going to be a multi-tissue analysis, so the collection of multiple tissues from the same patients in metabolic disease. And this will be from Panos and Johan on the StarNet cohort. And then finally, we're going to try to tie that all together and talk about how do we build all of this data into a complete single-cell epigenomics atlas, and can we use that data to predict functional non-coding variants using deep learning. And then last but certainly not least, we will have Melissa Thomas. Melissa it's a requirement to be the co-chair of one of these AMPs to have your name be Melissa. So Melissa was the co-chair of AMP type 2 diabetes, and she has kindly stayed on to be a steering committee member for CMD, and she is going to tell us about the trajectory ahead. So with that, I'm going to turn it over to Kyle for our first talk.
Great, thank you. And I'm happy to tell you about some of our work in generating, integrating single-cell data to annotate complex disease loci as part of AMP CMD. We've heard already talks today about single-cell genomics. Just to give a brief overview, so we can use single-cell assays to annotate gene regulatory programs in specific cell types. We generate profiles from individual cells, cluster the cells together to define cell types, and then we can identify pieces of information that can allow us to understand gene regulation in these cell types, for example, cis-regulatory elements, the activity of genes, as well as relationships between these elements and the genes they're regulating. I've listed some of the common assays that we use down below that many people, I'm sure, have also used to perform these assays, but there's many other single-cell assays as well that can be utilized to define cell type-specific gene regulatory programs. Within the context of AMP CMD, one of the primary goals is to understand targets using genetics and genes that are involved in genetic risk of disease. This is a challenge not just for metabolic disease, but for any complex disease, because most risk variants for disease are non-coding and require detailed annotation of the genome to understand what these variants are doing. So cell type resolved maps are quite useful for this purpose in that they provide information that you can intersect with genetic variation to annotate non-coding variants, to move from an association signal to the elements that they affect, the genes that these elements are affecting, and so forth, and gain an understanding of the genes that are targets of these variants. One useful aspect of non-coding variation as well is that it gives you access to information about the cell types, genomic regions, genes, and pathways involved in disease, as well as the context in which they're affected. So when and where are these genes affected, what cell types in response to what stimulus, as well as the direction of effect. Does increased or decreased activity of a gene increase risk or is protective for disease? So I will very briefly touch on several ways that we utilize cell type specific maps from single cell assays in combination with genetic association data to understand complex disease. So the first is identifying disease relevant cell types, annotating variants in these cell types. Second is mapping risk variant effects on gene regulatory programs in these cell types. Third is understanding how genetic risk affects changes in progression to disease or in a disease state. And last, as Naran and Melissa mentioned, how we provide access to these data from single-cell assays to the community through the AMP CMD portals. So for the first point, annotating disease-relevant cell types and risk loci. So I'll highlight one project here among many that we've done in recent years of defining basal maps of cell types in human tissues. This project was funded by the FNH and was work done with Bing Ren and Alan Wang and Josh, who's on the stage here at UCSD. And in this study, we profiled 45 human tissues and defined cell types from these tissues, 222 cell types in total, and identified over 1 million cis-regulatory elements across the genome, many of which had very cell type specific activity in one cell type relative to another. And one way that we can use these maps is by intersecting with genetic association data to identify cell types that are preferentially enriched for variants associated with different traits. And in doing that, we find hundreds of traits where there are very specific relationships between variants associated with that trait and sites active in certain cell types. I'm just highlighting a couple here amongst the hundreds of examples. For example, thyroid-stimulating hormone is specifically enriched for follicular cells in the thyroid. Atrial fibrillation is enriched for cardiomyocytes and so forth. So we find very specific relationships to many traits that enable us to understand the cell types that are involved in these traits and diseases, and then allow us to annotate variants that are overlapping sites in these cell types. The second way that these data were used is then going to individual loci, where you can take knowledge of the cell types that are enriched for a given trait or disease, and then look for variants that overlap these sites, and then predict not only the sites that are affected at these loci, but then also the genes that are targets of these sites. And I'm highlighting a couple examples here, one for ulcerative colitis and one for osteoarthritis, but there's many examples of very fine-grained insight into the likely gene targets of variants associated with many, many diseases. But these studies give us basal maps, meaning that they're just baseline maps of regulatory elements that are active in cell types in these tissues. In many cases, don't tell us about whether variants affect the activity of these sites, which is a further level of information which allows us to understand which cell types are being affected by these variants. So the next set of studies that we've done is to not only take variants overlapping sites active in certain cell types, but understand which variants actually affect these cell types. There's various ways to do this from single cell data that I've highlighted here, one of which is to use machine learning to learn sequence context for these sites, and then use what you've learned to predict variant effects on the sites, which is what Josh will talk about later on in this session. What I'm going to talk about briefly is measuring the effects of variants on cell type activity directly using QTL mapping. And so we again have done several of these studies. I'm just highlighting one here where we've generated single cell maps in pancreatic islets, as well as genotyping data from the same donors, and then we can correlate the presence of a genotype with the activity of a cis-regulatory element across individuals. And in doing that, that identifies thousands of sites across the genome that are affected by variant activity, a subset of which have effects that are specific to one cell type relative to others, highlighting the value of having these cell type resolved maps. And here's an example at the bottom here of a variant that is in a site that's active across different cell types, but only affects beta cells relative to the other cell types. And we can then use this information to annotate risk loci for diabetes in this case, given that it's pancreatic islets, and say here are variants that are associated with diabetes at this locus. These same variants are associated with genetic effects on regulatory activity in a specific cell type, in this case pancreatic acinar cells. And so it gives us a clue as to what cell type these variants may be acting through. It also gives us insight into the direction of effects. So in this case, the protective allele for type 1 diabetes is associated with a reduced activity of the site, giving us a clue that maybe reducing the site is protective for diabetes risk. So that's looking at changes associated with genotype and identifying genetic effects. The third way that we've used single cell data is to understand changes that are occurring in progression to disease or in disease states. So I've highlighted a couple examples here. One is a study that we've done in our lab looking at progression to type 1 diabetes across 33 donors, where we have single cell profiles generated in the pancreas. And we can identify pathways that are affected during progression to type 1 diabetes in individuals that are positive for type 1 diabetes autoantibodies who hadn't developed type 1 diabetes. And there are very specific pathways that we find both up and down regulated in these pre-disease states. For example, interferon signaling, antigen presentation, and so forth are upregulated in people that are progressing to type 1 diabetes. We can utilize this information, again, in combination with genetic variation to say these pathways are preferentially harboring variants associated with disease risk, as well as identify pathways that don't harbor genetic risk to distinguish pathways that might be in the causal route to disease versus those that are just consequential in disease. So for example, interferon signaling, antigen presentation enriched for disease risk, whereas processes like apoptosis and so forth are not harboring risk, and so may be consequences but not causally involved in disease. Another example of this is a project that's ongoing. It's funded by the FNH. where we're generating single-cell maps in the human heart. We have another project looking at single-cell maps in the human liver, which David will talk about next. In the heart, we're just starting to analyze these data. They're still being collected, but to give you some preliminary insight, these are data from 30 donors with heart failure or control, non-heart failing donors, and looking at changes in heart failure compared to control in each cell type finds dramatic changes in gene activity within many cell types, cardiomyocytes as expected, immune cells, and other cell types as well. But like I said, these studies are still ongoing, and the goal is, again, to intersect this with genetic risk to understand causal processes in heart failure. So finally, we work in close collaboration with Noel Burt and Jason Flanagan at the Broad Institute to help develop the AMP CMD Knowledge Portal, which comprises genetic data as well as genomic data, to enable people to be able to do these types of analyses themselves, to understand non-coding variants and what genes they're affected and what cell types and so forth. And we collect not only single-cell data, but a range of functional genomics data that's deposited in this portal. As part of these efforts, we make single-cell maps from public resources as well. So one example is PancDB, which is a public database funded by the NADK that provides single-cell data. And we make maps from these resources to then derive knowledge that can be utilized within the portal. For example, genes expressed in certain cell types, marker genes of cell types, changes in gene activity within cell types, so pieces of knowledge that can then be put forth on the portal to help people annotate risk variants and loci of interest. And then finally, the kind of overall goal of these portals is to provide, in one case, distilled knowledge, like I just mentioned, so pieces of information that are easily accessible to users, where they can look up a gene of interest or a variant of interest and so forth, and gain access to knowledge that we have about those entities, as well as interactive analyses that allow deeper exploration of the data, for example, single-cell maps and looking up the expression of genes within certain cell types or subsetting in various ways, as well as access to all the raw data. So thank you very much.
Thank you. I first have to tell you that the pointer works, and now that works. There we go. Okay, so the disease formerly known as NASH is now known as MASH. Sorry. Nothing else has changed as far as I can tell, but it stands for metabolic dysfunction-associated steatohepatitis. The concept is the same, though. A normal liver can develop steatosis, fat in the liver, which then become inflammation, fibrosis, MASH, steatohepatitis, and if this progresses, it can go on to cirrhosis and hepatocellular cancer. It's the most common disease by far in Western civilization. We've had the opportunity to receive livers that have been declined for transplantation, and the biggest reason they're declined is because of steatosis. Surgeons are concerned about primary graft dysfunction in transplanting a steatotic liver, so we've collected over 200 livers over the course of several years, which are available for this type of analysis. And much to our surprise, there's a wide range in steatosis, inflammation, and fibrosis in people who are believed to be healthy people, potential liver donors. Okay. In collaboration with Kyle and people at UC San Diego, we've started to analyze these livers using single nuclear RNA-seq and single nuclear TAC-seq. And the PCA analysis revealed that, as you expect, the patients are very heterogeneous, but if you look at the patients with MASH, with fibrosis, they tend to cluster together. So the people with normal livers and livers with just steatosis tend to cluster together, reflecting mild disease or no disease, while the patients with MASH cluster together, particularly those with more severe fibrosis. When we did the UPAP analysis, we found exactly what you would suspect. All the major liver types are present, in particular hepatocytes, sinusoidal endothelial cells, and hepatic stellate cells. The clusters did not change. The relative distribution of the clusters and the subclusters did, but the clusters did not change between the three phenotypes. We're going to emphasize here one of those clusters called the hepatic stellate cells. This is a normal liver where the UV light reflects the retinoids in the stellate cells distributed throughout the acinus. This is an electron microgram showing a stellate cell with its retinoid lipid droplet below the sinusoidal endothelial cells in the space of disc surrounded by hepatocytes. These are the cells that lay down the fibrous scar in MASH and in other liver diseases, other hepatotoxic liver diseases. They're normally the parasite. It's the only parasite in the liver that controls blood flow. But with activation, they start contracting. That's why they're called myofibroblasts when they're activated. They lay down the fibrous scar. And if you look at epidemiological studies, it's only the stage of fibrosis that correlates with mortality in this disease. It's not the steatosis, it's not the inflammation, it's the fibrosis. So therefore, we decided to first analyze these cells in this single nuclear analysis. Okay. The PCA analysis for stellate cells were, again, reflecting the underlying phenotype with the normal and the steatotics patients tending to cluster together and separated from the patients with MASH. These are the patients with inflammation and fibrosis. We believe that this is really consistent with the stellate cells being fairly normal when there's just steatosis. Okay. When we correlated the PCA and stellate cells, again, there was a correlation between the fibrosis score, MASH, and how they clustered, but it wasn't as tight as we expected. And we think what this reflects is that, unlike mice, the patient's stellate cells are much more heterogeneous. In other words, normal, healthy livers will have some activated stellate cells, and patients with MASH with fibrosis will have some quiescent stellate cells. This is not what we find in mice and shows the importance of studying this disease in humans. Okay. When we used our siRNA sequencing to look at the subcluster within the hepatic stellate cells, we identified 4 subclusters. The quiescent stellate cells, which are the markers normal liver, retinoid storing, happy stellate cells. Then there were 2 levels of activation. One was a frank myofibroblast, just as we saw in mice. The other was an intermediate stage, which we had not seen previously. And there was a clearly 4th subcluster, which represents inflammatory stellate cells. These are previously described in cancer, but not previously described in a non-cancerous liver. So 4 different subclusters describe the stellate cells. And when we look at the markers, these are the markers that we saw in activated stellate cells, whether in culture, in preclinical models, or in patient material. And these reflect an activated phenotype that's actively producing fibrillar collagen and other fibrosis. When we do a pathway analysis, we found that these major activation markers correlated with markers we previously identified, transcription factors that drive these. So a limited number of transcription factors drives the activated phenotype and produces these markers. And one, to our surprise, was a previously described protein we had previously described called LARP6. So LARP6 is a marker of activated hepatic stellate cells. What's LARP6? LARP6 we previously described as an RNA binding protein that drives the expression of the fibrillar collagens, alpha-1,1, alpha-1,2, and alpha-1,3. And it is responsible for the organization of the translation, the efficient translation, and the secretion. So LARP6, it was an interesting protein to follow up. To do that, we tried to develop a model of MASH in a dish. We took the exact same livers we had analyzed by single nuclear RNA-seq and single nuclear TAC-seq, and we took those cells, purified them, and reconstituted small spheroids, about 8,000 cells each. And we took those cells and incubated them in a MASH cocktail, so we're able to induce what looked like a MASH phenotype in a small culture of human liver cells. This is what it looks like. This is a spheroid. This is a spheroid with a MASH cocktail where you can see the steatosis, and even more important, you can see the expression of type 1 collagen, which is not present in the normal healthy spheroid. We then said, can we knock down LARP6 specifically using siRNA in hepatic stellate cells and reproduce the disease phenotype with or without the expression of LARP6? Here you can see the LARP6 knockdown was successful. And we look at type 1 collagen. It's greatly reduced in the LARP6 knockdown compared to the MASH disease in a dish. So a single knockdown of a single gene in a single cell was sufficient to disrupt the MASH phenotype in this model of human liver cells. You can see this a little more clearly on the RNA expression analysis of the same spheroids. Some of the markers of hepatic stellate cell activation fibrosis are down when we knock down LARP6. But more importantly, the production of collagen, the protein itself, is substantially down, as well as, to a lesser extent, alpha-synuclein muscle actin, which is a marker of stellate cell activation. So a single gene knockdown changes the entire profile of the spheroid and produces less type 1 collagen, which is the major driver of fibrosis. So in summary, we have performed single nuclear TAC-seq and single nuclear RNA-seq on normal MAFL and MASH livers with the goal of 90 livers. That's what Kyle tells me. 90, right? Okay. Thank you, boss. The donor level of PCA analysis separated the normal and MAFL livers from the MASH livers. UMAP clustering identified the 6 major cell types in the liver from the 3 patient phenotypes, all which are represented in all the phenotypes. The hepatic stellate cell PCA correlation separated the normal and MAFL, which is steatotic livers, from the MASH livers with fibrosis. Initial analysis of the hepatic stellate cell cluster revealed a quiescent, an intermediate activated, an activated, which is the same as a myofibroblast, and an inflammatory subcluster. One of the things that fell out of the initial analysis is this RNA binding protein LARP6, which identifies the activated hepatic stellate cell subcluster. And then I just showed you knockdown of LARP6 in the human liver spheroid model of MASH blocked fibrosis, the production of type 1 collagen, and thus the functional role of genes identified by multi-omics can be rapidly assessed in human disease models. Thank you. Thank you for attention. Right on time.
So my wife's name is Whiteman, and before we married, she suggested we would take that name, and I kind of regret we didn't, because Bjorkegren is hard to say. Anyway, so let's, okay, I thought I had this. Is it the middle one? Sorry? I push hard. Oh, the right one. Okay, good afternoon. I will talk about tissue and cell level studies in human cardiometabolic disorder and coronary disease, and more specifically the StarNet study. I will start with the tissue, and then Panos will tell you more about the single cell studies we're doing right now. So StarNet is a study which initiated in Stockholm. I had a short career as a heart surgeon, and then I started a collaboration with Arno Rusalep, who since 2007 have gathered tissue samples from patients undergoing bypass surgery, as you see here on the slide. And since then we have gathered nearly 1,500 patients and nearly 500 controls. And importantly, these controls are, as it says here, coronary disease free, as they are preoperatively examined for not having the disease, which is unique compared to Jiva studies where you have somewhat contaminated controls. I was recruited to Mount Sinai, and together with Eric Schad, we generated a lot of the data that we show here on the table, which now are available to a large extent on the AMP for common metabolic diseases, including bulk RNA sequencing from these tissues, but also whole genome sequence, and some plasma proteomics and metabolomics in these cases and controls. Luckily, these GWAS risk loci didn't end up in coding regions of genes, so it was when we started to analyze StarNet, one of the major tasks was to define genes responsible for genetic risk loci, and we published 50 or 100 of those articles using StarNet. But maybe the one more important one was in Science, where we actually showed that cardiometabolic risk loci share downstream cis and trans gene regulation across tissues and also diseases, and many of those diseases that are part of this AMP program. But really, my interest is gene regulatory networks. So recently, we published a paper in the inaugural edition of Nature Cardiovascular Research, a mechanistic framework for cardiometabolic and coronary artery disease, which consisted of hundreds of networks across these tissues that were associated with both cardiometabolic and coronary artery disease. And before I hand over, so basically the goal for us is to use these new systems approaches to marry them with the pathway approaches, which we are not really there yet. But that's why I'm so excited we now have funding from FNIH and the AMP program to run single nucleotide data, because this is one of the examples of the bulk RNA-seq network that we found. It's an atherosclerotic arterial wall gene regulatory network, and as you can see, it contains a lot of these risk genes identified by GWAS for coronary artery disease. It actually contributes markedly to heritability of coronary artery disease. But there are also these genes that were mentioned in early talk that we call key drivers that are a high hierarchy affecting this network heavily. And I think my kind of a goal is to make the pharmaceutical industry more interested in these key drivers, because if you perturb them, you get a huge effect on the network and associated phenotypes. So I think the end goal is to get this molecular map using networks across tissues down to the cell level, so we can really understand what's going on instead of focusing on individual successes like GLP-1 or similar, where we are happy about the outcome, but we really don't understand what's going on. So I think we need these networks mapped. And this particular network was affected by LDL cholesterol. So we think levels of LDL actually affect this network, which are modulating extracellular matrix, which in turn affects syntax score, atherosclerosis. So I just want to, since Francis Conley was here this morning, we were very happy to be acknowledged by the director blog back in the days, but I guess he's not here anymore. So by that, I hand over to Panos, and he will talk about the latest single nucleotide data. Thank you.
Great, thanks so much, Johan. So just to summarize that the goal of working with Johan, the goal of this project is to generate single nucleus multiomics data within a subset of the StarNet cohort. So for that, we're using the commercial available 10x kit for the multiome. Now, in terms of the cohort, we're actually using a small subset of the StarNet. So we have some what we define as CAD cases. These are cases that they have surgery for atherosclerosis. And then we also have controls, and these are controls that they have surgery for other reasons other than atherosclerosis. So most of the time, this is for replacement of some of the valves. And as you can see here, we have a very powerful design, because even within the cases, we can actually look for atherosclerosis, which is within aorta. But then within the same individual, we also have a healthy vessel from mammillary artery that we can use as a within subject control comparison. And then also we can do the same analysis by looking across donors, right? Because we have aorta from a control that doesn't have atherosclerosis. In terms of the cohort, we try to keep a balance between males and females. And then in terms of the age and the BMI, the case and controls are very well matched. And overall, we are supposed to generate a single nucleus multiomics data from 260 overall dissections. So this is the timeline. I just want to emphasize that we just started our second year, so we are kind of a new project within the AMP CMD. And in terms of the timeline for the single cell data generation, we're actually supposed to be happening right now. One thing was that as part of the AMP CMD consortium and working also with many other different groups, as part of working groups, we realized that there's a need to further explore the way and the methods that we actually utilize to generate the single nucleus multiomics data, specifically how you isolate nuclei from different tissues. So for that, even if it wasn't planned to do a pilot study, we ended up actually need to do a pilot study to make sure that we have the best practices. So for that, we actually used the best protocol for generating single nuclear RNA or ataxic data from existing publication. We also discussed this with Parker Lab. They have a really nice protocol working directly with muscle tissue. And the idea was to compare this kind of best practices with a commercially available kit coming from 10x. And the goal here is to be able to provide a simple protocol that every lab can apply, so you actually have a better chance for standardization of data generation, so it's easier to harmonize data across different labs. And at the same time, we would like to have a single protocol that when we generate data across different tissues, this is harmonized as much as possible. So for that, we compare head-to-head the best practices with the commercial available protocol. No need to go through here all this slide, but after we did all this kind of pilot study, we realized that using the 10x nuclei isolation protocol is working very well, and it's performing equally well with all the other kind of best practice protocols that we have. So this is the one that we actually put in the pipeline, and we already have generated in other libraries, and I believe the sequencing should be done by now. And the way that we do the design, we care about minimizing the cost, while you can also reduce the technical batch effect. So we have developed a protocol that you can pull different individuals together in the same kind of single cell library. You can have multiple libraries per pool, so you can have actually technical replicates. And then by multiplexing individual, you can reduce the batch effect and then use the genotype to be able to do the demultiplexing. And this is something that's working extremely well, and we have already performed this kind of data generation. Again, I want to thank Johan, who is the PI for this study, and there's many other people in my group and Johan's group that they have participated. Thank you so much.
We'll figure this out eventually. All right, thanks everyone. My name is Josh Chu. I'm a geneticist at Pfizer, and today I'll show you how we're combining human genetics with single-cell epigenomics and deep learning for drug discovery. And so in the last few years, there have been numerous genetic studies, or genetic discoveries, primarily enabled by GWAS of large-scale cohorts, such as the UK Biobank. But I think to date, GWAS has largely been untapped for drug discovery because of the challenges in understanding the genetic mechanisms of non-coding variants. And so for drug discovery, some of the key questions that we ask are, what are the causal genes of these non-coding variants? What cell types do they act through? And most importantly, what direction do these variants have on their causal genes in disease? And so the prevailing thought is that non-coding genetic variants mediate disease risk primarily by affecting gene regulation. And this is often through cell type dependent context. And so a non-coding variant could disrupt the binding site for a transcription factor at an enhancer, leading to altered enhancer activity, downstream effects on gene expression, and then ultimately impacting disease risk. One of the ways that we've used GWAS for target discovery is by combining it with single-cell epigenomics. And so in this example, there's a non-coding variants associated with kidney function at the DPEP1 locus are likely acting through proximal tubule cells in the kidney, and through allelic imbalance, we're able to show that this locus is consistent with inhibiting DPEP1 in order to increase kidney function. But of course, for drug discovery, we're not just interested in the kidney. And so what we'd really like to do is ask these same types of questions and apply the same type of analysis to all cell types and all tissues in the human body. And so allelic imbalance and QTL-based approaches are great, but they're limited if we want to try to ask these broader questions. One approach that we and many others in the field have taken is to train deep learning models on single-cell epigenomics data to try and predict the effect of non-coding variants. And so there's been a lot of work in this space, but recent methods such as Informer train on even larger regions than previous models, giving these models more regulatory context. Another improvement is training on and predicting continuous epigenomic signal, which improves the interpretability of these models. And so together, I think these two advances have really improved variant effect predictions. But to train these deep learning models, we need a lot of data. And so one potential source of data is from single-cell human epigenomics reference atlases. But even the largest and most current reference atlases are incomplete. They're mostly based on tissues from a few healthy donors and likely underrepresent or miss certain disease-relevant tissues, such as the eye, kidney, and lymphoid tissues. Another major limitation is that rare cell types and subtypes are not well resolved in these epigenomic atlases alone. And so new methods that use deep learning such as multivi, can integrate multiomics together with single modality attack-seq data for increased cluster and cell type granularity. And so to create a more complete single-cell epigenomics atlas, we took a look at the published landscape. We saw that there were over 1,700 publicly available single-cell attack-seq and multi-omic experiments. And these come from large atlas resources such as AMP and ENCODE, as well as consortiums that focused on profiling a single tissue in depth, as well as many publications focused on a single tissue in one disease. But to our knowledge, there's not been a coordinated effort to reprocess and compile all of these experiments into a single unified resource. And so colored in blue here are all the new experiments that aren't contained in the current reference atlas. And so you can see that for certain tissues, such as brain, heart, and kidney, and pancreas, there's a lot of new data that we could use. These experiments span multiple technologies, and around a third of them are multiomics with paired RNA and attack-seq readouts. But of course, to combine all these experiments, we need extensive quality control, especially because these experiments come from many different labs, different tissue sources, and different technologies. And so we have filters on the metadata level, even before clustering and after clustering, to ensure that we're removing low-quality cells and potential doublets to make sure that we have the highest quality starting point to compile this resource. And so after all these stringent quality control steps, we were left with around 7.5 million cells from over 1,500 experiments to build our single-cell epigenomics atlas. And so we used multivi to combine all of the single modality attack-seq and multi-omic experiments onto a single UMAP embedding. And at a clustering resolution of 1, we identified 61 broad cell group clusters. And after subclustering each one of those main clusters, we obtained a total of 401 cell type subclusters. And so I'm showing an example here of that subclustering process for cluster number 29, which corresponds to bipolar cells from the retina. So on this UMAP plot, you can see that each one of these subclusters is visually distinct. And because we have a multi-omics atlas, we're able to use the observed gene expression from the multi-omic data, as well as imputed gene expression data from the single modality attack-seq data, in order to label each one of these subtypes by comparing them to previously published single-cell RNA-seq studies. We also confirmed the presence of lineage-specific, as well as more subtype-specific chromatin accessibility around promoters of these same marker genes. So with this single-cell atlas, we then wanted to combine it with genetics in order to understand potential safety risks from genetics. And so one example is at the FGF5 locus, and at this locus, there's a protein QTL from the UK Biobank, where the variant results in increased circulating FGF5 protein in plasma. The same GWAS signal is also associated with increased kidney function and decreased risk for chronic kidney disease, but is also paradoxically associated with increased risk for cardiovascular disease and increased blood pressure. And so we hypothesized that this was perhaps due to two distinct genetic mechanisms acting at this locus through two different cell types. And so when we took a look at the genetics for this locus, and we looked at kidney function and cardiovascular disease, that's exactly what we saw. And so one variant for kidney function was specifically active in enhancer for proximal tubule cells in the kidney compared to all other cell types that we had data for. And similarly for AFib and other cardiovascular diseases, we saw a different variant that was active in cardiomyocytes in the heart. And so this could potentially explain why we see these paradoxical associations for renal and cardiovascular disease. And so finally, we wanted to make use of our epigenomics resource to predict the effects of non-coding variants. And so I've been working with Joran Rittel, who's in our machine learning group based in Berlin, and he trained deep learning models based on informer architecture and showed that he was able to predict epigenomic signal from DNA sequence alone, and that this predicted signal was highly mirrored the observed signal from the actual data. And then similar to previous publications, he then calculated variant effect scores by taking the difference between predicted epigenomic signal between the two alleles of each non-coding variant. So in order to scale up these sorts of deep learning models and train on even more data, we also need engineering solutions. And so Jorn and the machine learning group have also been working on open source Python packages such as Bigwig Loader that massively speed up batch loading for loading these single cell epigenomics tracks and training these deep learning models. And so in summary, today I've shown a more complete single cell multi-omics atlas that spans multiple tissues and multiple diseases for a more complete picture of genetic mechanisms for drug discovery. And we hope to make this atlas available to the general public as soon as it's ready. At Pfizer, we're also working on open source methods and packages for training deep learning models and working with large-scale single-cell epigenomics datasets. I want to acknowledge all of my colleagues, especially Joran Rittel, who've contributed to this project, and we also want to acknowledge all the authors of countless publications who've made their datasets publicly available and that I've included in this resource. Thank you.
One more. So it's my pleasure to be the last part of this series on our Common Metabolic Diseases Accelerated Medicine Partnership Project. And great pleasure here to talk about the trajectory ahead after so many great presentations. So as we were framing this project in 2020 and 2021, coming off of the successes of the Accelerated Medicines Partnership for Type 2 Diabetes, we understood that moving forward for looking at how to build new therapies in common metabolic diseases, we needed to move away from the siloed approach because our patients often experience multiple common metabolic diseases over the course of a lifetime, and that these are collectively major challenges for public health, and that historically, therapeutic interventions primarily targeted single diseases. And so the opportunity to modify multiple metabolic diseases outcomes by addressing common pathophysiologic drivers was very compelling. But we understood that we needed a different kind of project that would move beyond single diseases. And as we were looking at these patient populations, it was really well known in the clinical environments that individuals living with obesity over time had a higher risk of developing type 2 diabetes or coronary artery disease, or if you started from the lens of type 2 diabetes or even pre-diabetes, you could watch the progression of multiple diseases, and many of our patients would be multi-medicated by the time they reached middle age. So with that in mind, we were in a very special position. There was a very innovative infrastructure in AMP type 2 diabetes with the knowledge portal that had been built. It over time had built a series of analytic tools and capabilities as well as a broad range of data that gave us an opportunity to take this and scale it across a larger number of diseases. And as Melissa showed you, this Common Metabolic Diseases Knowledge Portal over the last several years now has a large number of data sets across organ systems related to these diseases and phenotypes in a wide range of areas. This gives us an opportunity to combine that information about genetics and clinical traits with a number of analytic tools so we can ask questions looking for those targets that might impact multiple diseases in a very different way. And as Kyle showed you, with the Common Metabolic Diseases Genome Atlas, we get the opportunity to integrate tissue or cell type information. So within the AMP T2D Knowledge Portal, Let me just show you a few of the types of tools that have been built that allow you now to ask these types of questions. So let's say you're starting with a gene of interest that you think might be a potential therapeutic target, but you really want to understand where might it work, in what traits or what disease types. The Huge Calculator is a tool that allows the integration of both common variant associations as well as rare variant associations from larger, more deeply genotyped cohorts. And together, using Bayesian statistics, can create a score that gives you a higher probability around gene associations. Now, in the portal, you have at your fingertips within a matter of perhaps even less than a minute to see a PheWAS visualization of your gene across multiple traits, as you can see visualized here. And although I know this is small on the screen, what the color coding bars show you at the bottom is actually, are you looking at cardiovascular trait associations? Are you looking at renal associations? Are you looking at hematologic associations? So very quickly, you can see both what you were looking for, but maybe what you really should know as well about other phenotypes that might impact your patients. Now, in addition to that, you can also look at the visualization in tabular form. So if you really want to now get down to the details, what you can see is which of those traits had the strongest statistical association with this gene based on the Bayesian huge score. And those with the bright green boxes have really strong statistical associations. And now you can start taking a look at that and say, with ApoC3, I think I need to take into consideration renal associations, lipid associations, and you begin to dive more deeply in the portal. But let's say instead that you were really interested in looking at a custom set of gene-level associations. And so now you're going to ask a different question. You're going to say, well, I wonder what are the genes that would be associated with risk for a high BMI and type 2 diabetes if I were really wanting to modify both the diabetes phenotype as well as the obesity phenotype. This tool, with the gene sifter tool with the MAGMA algorithm, will allow you to ask those kinds of questions and very rapidly leverage the huge algorithm to see what goes to the top of the list, what are the genes that are of interest. But that's not all you can do on the portal. If instead you've come up with a trait-based association or disease-based association with a particular genetic variant, and you really want to know what else is that variant associated with besides the paper I read last week that said it was really cool for an esoteric disease. Well, perhaps you want to understand a different question, not just that variant, but if that variant was associated with increased fasting insulin, well, what if I wanted to look for a target where I could actually decrease insulin resistance, improve dyslipidemia, and increase HDL cholesterol? In that context, you can put multiple traits into the variant sifter tool. And there, you can see which variants come to the top and also the directionality of the effect on these traits that you're looking at. So now you have a group of genetic variants that might have been interesting rather than the one that you read about that had been highlighted in the literature. And you begin to see, I can go deeper in the portal with respect to these particular variants because maybe this is a thread that I could pull to look for opportunities to modify both insulin resistance and dyslipidemia. It's just an illustration of how very quickly even someone who's not a genetic epidemiologist can ask some questions for hypothesis generation across multiple common metabolic diseases. But as you've heard from Kyle and others, many of these genetic associations that we find are in non-coding regions, and it's difficult to know what the target gene is associated with a particular genetic variant once you've built a list like this. And so this additional target prioritization workflow needed to move from the prioritized variants to variant-regulated genes and then to the potential targets. And that's really where this aspect of the portal and the project integrates with what you've seen on the work on single-cell and epigenetic data generation, both in healthy tissues and in diseased tissues. So within AMP CMD, we've been working to generate data in many of these gaps, particularly filling the gap in terms of disease versus healthy tissues. So this is an example from a beautiful set of work from multiple investigators, including Francis Collins, Laura Scott, and Steve Parker, that really leverages a deeply phenotyped human cohort, the fusion cohort, that has a broad range of phenotyping, including glycemic phenotypes, metabolic phenotypes, biomarkers, clamp data. And in this particular cohort, skeletal muscle biopsies had been collected. And so, in the context of a very deep set of phenotypes, single nuclear ATAC-seq and single nuclear RNA-seq data have been generated. Very much like you've seen in the workflows from other tissues, the cell types have been identified, and understanding now at a cellular level where the epigenetic marks are that may be associated with signals that could be quite important associations with insulin resistance or with other aspects of skeletal muscle dysfunction. And Steve and colleagues have recently publicly put this paper up in preprint form to show you the depth of data that will be coming in over time for queries in the portal. Another example is to look at a different deeply phenotyped cohort. This is coming from Karen Small in King's College London. This is the Twins UK cohort, where adipose biopsies, as well as circulating metabolomics, were conducted. And what Karen can do is she can annotate these non-coding variants, not only with circulating biomarker data, but with the EQTL signals associated in adipose tissue. So connecting the dots between things we can measure in blood in patients with what's happening in the primary tissue and cell types, and then back to the genetic variants that are of interest and at risk. So as we look at the trajectory ahead for AMP CMD in the next 2 years or more that we have ahead, we're continuing to expand genomic and epigenomic data from deeply phenotyped cohorts that will support our functional annotation of these non-coding variants and our ability to take a variant to a gene to a target. You can see across the top the common metabolic diseases, and that we were prioritizing cohorts where we can compare healthy tissues to diseased tissues, because as Kyle pointed out, many of the epigenetic marks may be quite different in a disease context as they will in healthy tissue. And so both from the NIH and the Opportunity Pool Awards and from the FNIH and the RFPs, we'll be expanding the data resources to support target prioritization. But at the same time, we'll still be building more tools in the Epigenetic Atlas and in the Knowledge Portal, so that we can see, just as we did in AMP type 2 diabetes, that as the scale of the data grew and the quality of the analytic tools grew, the insights and hypotheses that we could generate leveraging these data began to really amplify in a synergistic way. So with that, I'd like to turn it back to Melissa with some interesting announcements.
All right, before we take questions, as Melissa just alluded to, we have generated quite a lot of data, but we've got about 2.5 years left on the project. And there are certainly some diseases and tissues where we would like to have more data or where we haven't really yet collected anything. So just a couple of days ago, we announced 2 new requests for proposals. And these are somewhat similar in nature. Both of them are around the generation of multi-omics data from both diseased and healthy tissues. What's a little bit different than what we've done in the past is that for the first one, we're really focused on, or we will give highest priority to tissues of diseases that we are not currently well represented in the project. And for the sake of this all hitting on one slide, I didn't list anything up here, but the link to the RFP is right there, and please feel free to come talk to either Neuron or I at the break if you're interested in learning more. And the second one, of course, as we all are in this area, very interested in being able to start generating, or at least generating more data in populations from non-European cohorts. So we have a specific RFP out for that, where we would be looking for tissues in diseased and non-diseased subjects, as well as corresponding genetic data that would go with that. So with that, I am happy to use any of our remaining time for questions from the audience. Thank you. Any questions from the audience? And for people on the stage, the mics are push to talk, and only 3 will work at a time.
Hi, Gil Oman again. So for many reasons, it's a common practice to try to figure out what are the targets that would be the sites of efficacy and what are the targets that would be off-target toxicity. And I just wonder if these portals, these data sets, seem logical to be used for such purposes if they are being used with those kinds of questions. Maybe from Joshua or Melissa.
Sure, I can take that question. Yeah, so as I showed in my talk, we're already thinking about this for target discovery. When we're evaluating new targets, we look at the genetics as well as the epigenomics to try and understand if there's potential safety risks of targeting a certain gene or a target or pathway for disease. And so, yeah, to answer your question, that is something that we're already looking at, and I think in Noah's talk, she might be able to give you a better picture of how the portal can be used for those kinds of questions.
And I'll add to that. So I think the FEWAS view that I showed you is one that's a beautiful view for hypothesis generation, both for efficacy and safety. So when I am in a meeting and there's a new gene that we have someone really excited about, I'll often pop it up in the portal and take a look at what I'm not hearing in the presentation. I may be hearing that it's a wonderful therapeutic target for a particular indication, but what else does that gene regulate? And I think FEWAS is only the beginning of a hypothesis, but it tells me the other disease areas to look into or the other mechanisms related to the gene. And that might even mean that if I have a tool molecule in a preclinical discovery program, I will test some biomarkers or other functional consequences that might be on the safety side of activating or inhibiting that mechanism. I'd say the other thing that we didn't highlight in the presentations but will become much more important over time is that many opportunities now for therapeutic interventions may be cell-specific with delivery technologies and genetic medicines. And so understanding from a mechanistic standpoint where the signal is, what the cell type is, could entirely change your approach to building an opportunity to really interrupt a disease. So I think tools like we are building will be very helpful in efficacy and safety and even understanding mechanistically what you would need to do to build a new therapy that might really modify a disease.
Sounds great.
Rima Dauk, Duke Medical Center. Fantastic presentations. As we think about metabolic disorders across Foundation NIH interest and programs, and I think about the diabetes in this case here, you know, thinking about lifestyle, diet, the exposome, the gut microbiome, there is so much that influences health on top of the genome. So I worry a little bit about focusing too much on the genome, which we should focus on, without paying attention to a huge array of other influences that modify your, the epigenetics and on and on. So I wish we can create a dialogue also as we build these resources for Alzheimer's to think about diabetes and others.
Yes, I think that's absolutely a great point, you know, that we really need to be collecting not just phenotype data on disease, but also better phenotype data on lifestyle as well. Any other questions? I realize we started late, so I have no idea how much time we have left, so I'm waiting for somebody. Okay, Courtney's giving me the 5 minutes. All right, if there aren't other questions from the audience, I'm happy to take chairperson's privilege. I had a couple for the speakers. Actually, one I have for Johan or Panos, and I think this is, you know, broadly applicable to a lot of AMPs. In common metabolic diseases, we often are looking at people, as Melissa said, who are multiply medicated with multiple comorbidities. Your cohort is really unique in that you have tissues, multiple tissues from the same person. I think it's really kind of a holy grail, and it's something we really talk about, you know, wanting to have in other cohorts, and it's just so hard to find. Can you give us some insight? How were you able to sort of go about collecting this cohort? What were the challenges and what made it successful?
Thank you. I just comment on the earlier question as well, that we actually have feces sample and we're generating microbiome in StarNet as well. So, yeah, I mean, it is not easy, and as I kind of alluded to, I had a short career as a heart surgeon in order to get into the surgical hall to get these samples. And I mean, we present all these sophisticated analysis downstream, but it will never be better than the initial biopsies you're taking. And a big advantage with StarNet is that our living patient, and we can have 5-year follow-up data, and we had taken so much effort to get really high quality RNA and tissue samples. And we're focusing on a limited number of analysis, because there's also kind of a problem with this, that you may be aiming for too much with histology. We want to do too much with the tissues, but we try to really get high quality data. And I think we outperform GTEx. I think one reason for that is because it's a disease-focused biobank, and I think those are really important to understand specific diseases, but also because the quality of our samples. And Melissa, it takes time, and I think more effort about what are the other StarNet-like biobanks that we need to generate, functional data, because there is a lot of focus on the genetics part of disease, and that's clearly important, particularly in early stages. But in the later stages, we need to understand the environmental factors, the disease factors, the age factors, and they will surely integrate with genetic variants, but there will be genetic variants that are silent most of their life, but when you kind of are exposed to certain environmental exposure, they come into action. And I think these more environmental-driven targets, like perhaps these key drivers, could be, I'm pretty sure they will be very interesting targets to consider in the future.
Limited, and one cannot embark on all, but to be thoughtful across the foundation programs to at least collect the samples, maybe in a standardized way, be thoughtful about what more can be done perhaps later. It cannot all be done up front, so that we can crosstalk, especially metabolic disorders, and many of these are metabolic disorders. We can collect in the right way, maybe standardize, harmonize, keep resources available, so the big investment can be leveraged later on.
Definitely, and I think I really look forward to the strategies for integrating the data from different disease phenotypes, because as I mentioned, StarNet liver is for some reason highly enriched for signal of Alzheimer's disease and inflammatory disease, and also, so I think leveraging these different AMP programs and integrating all the data, that should be kind of a task for Kyle and others to take on, and I think that's where we really can have the big synergies.
All right, I think Courtney is giving me the polite head nod, so I think with that we will go ahead and close this session, and if I'm actually, I don't want to pretend I know what's going on with the agenda. I was going to suggest we have a break, but I'm going to let Courtney say.
Yes, we do have an actually scheduled break on the agenda next. So thank you to the CMD team for such a great session.